Okay, I just start by Assalamualaikum, good morning, students and Prof. Sharif. We are very honored today to have my mentor, somebody I look up to, Prof. Sharif, to give you guys two lectures. So today will be ectoparasite and next week will be endoparasite. Without further ado, I will pass the stage to Prof. Sharif.
Okay, thank you, Dr. Diana. You can see my slides now?
Yes, Prof.
Okay, full slide. Okay. Good morning, everyone. Assalamualaikum, and hope you all are keeping well. Today, I was requested to cover diseases caused by ectoparasites. Now, before I go on, I'd like to introduce myself, a little bit brief introduction of myself. I don't think I've taught this class. So, short, brief introduction. I was born in Seremban. I went to school in Perak, Tapah. I did my DBM in Pakistan, Faisalabad, and my PhD and masters in University of Stirling in Scotland. You all must have, some of you must have seen the movie Braveheart, the Wallace Monument that is in Stirling. You can see from, I was staying in a residence hostel, and you can see the Wallace Monument where the sword is kept. I joined UPM in 1976, more than 40 years ago. All right, now I'm retired. All right, okay. Now, normally I start my lectures with a quote for the students. So I have many quotes. For a quote for today, I would like to share with you. Before you speak, let your words pass through three gates. So before you say anything, you must pass through three gates. That is it true what you're going to say? Is it necessary what you're going to say? Is it kind enough what you're going to say? Very important. In daily life, when you go about, you talk to your colleagues, you talk to your friends, you talk to your parents, make sure what you say passes through the three gates here. Is it true? Is it necessary for you to say that? You may hurt the feelings of the other people, So be kind, be wise, very important in your daily life. Okay, that's a quote for today. Let's continue with the lecture today. All right, ectoparasites. Ectoparasites are the most common cause of fish mortalities. Of course, the virus are important, the bacteria also are important, but of course, the ectoparasites are very important as well, right? Especially in larvae and fingerling stages, right? Very young stage. In elderly fish or advanced stage fish, it's not so severe as in the younger fish. Why they are so common in cause of mortalities? Because they're very rapid in terms of multiplication and so on. Doesn't involve a secondary host, the direct, all right? And these ectoparasites are the protozoan group, the crustacean group, and the monogenetic trematodes. So there are three major groups that form the ectoparasites or cause ectoparasitic diseases in fish. All right. So I'm going to first cover with you the protozoan parasites, right? White spot. Why do they call it white spot? You can see the clinical sign on the body of fish. It's white spots. What are these white spots? These white spots I will explain to you more detail. The parasite goes and lodges underneath the epithelial cell, not the skin, just the surface epithelial cells. So that's why they call ectoparasites outside the skin. All right. So what causes white spot? The parasite name is very long. It took me about 2 months to say the name correctly. It's Ichthyotherius multifilis. Don't worry if you cannot say it properly. All right, there are two versions of this parasite, or two forms of these parasites. One is found in the freshwater fish, and the other is found in marine fish. So the marine fish is different, the name is different. Freshwater is Ichthyotherius, and marine is Cryptocrayon irritant. So it's worldwide in distribution. It's in all fish species. It's not to specific host. All right, and it's found. That means it is found in food fish, in aquarium fish. All right, and it's associated with rainy seasons. Usually, when it starts raining, the temperature drops by 1 or 2 degrees Celsius. Now, when the temperature drops, it then takes advantage of the stress condition of the fish and it infects the fish. I'll explain to you a little later. Yeah. All right. So this is the shape of the parasite, protozoan parasite. You can see the cilia all around the body. You can see the macronucleus, horseshoe shape. All right. And granular cytoplasm. It looks like sand. Right. So these are the typical features. Granular cytoplasm, horseshoe-shaped macronucleus, and is round in shape, of course. You can see here the horseshoe-shaped nucleus. There are several of them here, all right? So this is 1, 2, and so on. So you can see there are different sizes also. It depends on the developmental stages, all right? And they they are round, they turn like a bicycle wheel. If you find them in your, I will show you how to make a diagnosis for this. You just make a fresh smear and you will find them moving like bicycle wheel. So they move around, right? The cytoplasm is granular, as I explained. Macronucleus is horseshoe shape, cilia all around the body, and they measure up to 1 mm. So this is the life cycle. They are found underneath the epithelial cells, and when they come out as mature forms, they call trophons, right? And they form a cyst, and in the cyst, they start dividing. You can find up to 200, 400 um, young cells in the cyst. And once they are ready, the tomites, we call them the tomite, they break the cyst and they find a host. So very simple life cycle. You can see there is no intermediate host and dies within 48 hours at this temperature, right? That means if you vacate your tank, you keep your tank empty for 4 days or 5 days, they will all die. So that's one way of treating or killing this parasite without the use of any chemicals. Just keep the tank empty. This is for the aquarium tank, of course. You can't do it on a big pond unless the pond is empty. All right. What's the clinical picture of fish infected with this disease? It's anorexic, doesn't eat. You find the white tubercles, we call them tubercles. And the cornea may also be whitish. I'll show you some pictures. Erratic swimming behavior. All right. What happens is when the fish, normally you would see in a tank, fish swimming were in a calm manner. All right. uh, in this case, they will start erratic. That means suddenly swimming as if there is something on the body they're trying to get rid of, right? Showing that the body is itchy. Now, they don't have hands, they cannot itch like what you do when you bite, mosquito bite, you do that. Fish cannot do that. So they do a swift movement to try to get rid of the parasite. So that's called erratic swimming. Or they rub against the side of the tank and they come to the surface for oxygen. I'll explain to you why. So the additional clinical signs, fish group at the inlet. Why at the inlet? Because there's more oxygen. The water is fresh. Later stages, when they become weak, they get swept by the current to the external part of the pond where the water goes out. They're lethargic, they don't move much. And the mortality is in the night because in the night there is low oxygen in the pond. In the day, there is photosynthesis because there is chlorophyll in the pond, algae, But in the night, is the reverse. Carbon dioxide is higher, oxygen is low, and the fish cannot breathe properly, and they start dying. So you can find the whole crop dead within a few days, within a week, if you don't take care, right? If you don't treat, okay. So this is to show you the eye, and you can see the excessive mucus. All this is excessive mucus, and you can see the white spot here. There are many white spots here, right? All these are parasites underneath the epithelial cells, right? And they're growing, becoming matured, right? And you can see a lot of these white spots here. So each one is one parasite, each dot, right? And you can see on the skin here down, further down. Sometimes it's difficult to detect if you are not experienced on a silver color fish. It may be difficult to see, but with your experience, you can detect it, right? Right, you can see the gills here, heavily loaded with the parasites. Of course, in this case also, there is excessive mucus. And when there is excessive mucus, fish cannot breathe properly. That's why they die in the night when there's low oxygen. And that's the other reason why they come to the surface to take in oxygen from the air. You can see them here. So this is, I wouldn't say this is white spot. It could be white spot or it could be other diseases that causes damage to the gills and they come up to get oxygen from the water surface. Now, how to diagnose the case? All right, you do scraping. First, of course, when you get the fish, you, of course, Dr. Diana will do a practical class with you. You do first spitting to make sure the fish doesn't jump around. That means you cut the spinal cord with the scalpel, and then you can use the same scalpel to scrape the mucus and all the gills to scrape the mucus. Here it is, and you put it onto a cover slide, and then you see under the microscope. So this is fresh smear. Of course, you will do a practical class on this. So if you do histology, you will find the parasites under the epithelial cells, right? So that's the skin, dermis layer, and this is underneath the epithelial cells. So you can see the horseshoe shape macronucleus and the granular cytoplasm. Here's another one. And there's another one there. Okay. And this is to show you how the gills are affected when the fish is infected with this Ectotherius multifilus. And you can see the parasite lodge in between the filaments. Again, the epithelial cell of the filament. You can see many of them. Some are young stage, some are bigger stage, advanced stage, and there is hyperplasia. So when there is a lot of hyperplasia like this, the fish cannot breathe and the fish dies of suffocation. This is what the normal gill looks like. You see, it's very clean. in between. So this is the primary lamellae, and this is the secondary lamellae. I'm sure you'll be taught in the practical class, or you may already have attended the practical class. And let's look back at the hyperplastic cells. See, so they block the exchange of oxygen, and that kills the fish. And that's why you get more mortalities in the night, right? And that's the other reason why they gather at the inlet where water is pouring into the pond. Right. So here you can see, of course, these are different, just to show you the single layer epithelial cells in the gills, in the secondary lamellae. And the parasite will be lodged just underneath that. So it's very delicate structure, the gills. And when you have a lot of these parasites, of course, it will kill the fish. So what's the treatment for this disease? Of course, first thing, you reduce stocking densities. Higher stocking densities causes stress to the fish. Of course, the bigger the number of fish in the pond, the faster the parasite will find the host. Increased temperature. Remember, I told you when there's rainy season, the temperature drops by 1 or 2 degrees, and the parasite takes advantage of fish under stress, and they multiply faster, they infect the host. Right? So you can increase the temperature by 1 or 2 degrees, not in the big pond. It's not possible. In a tank at home, yes. So this I'm referring to is in an aquarium tank or in a tank in a hatchery. Right? Now, use formalin. Yes. Formalin is used for treatment in fish, but in very low dose. Right? And you can use methylene blue. There'll be a lecture for you by the other lecturers on how to treat fish, right? And we use bath treatment. They'll explain to you how bath is given. And for methylene blue, you got to add 1.1 because it breaks down fast. So you got to add additional on alternate days. That means you add Monday, the first dose, Wednesday, another dose of 1.1 ppm, parts per million. You can use sodium chloride as well. The salt, table salt that you use at home, you can use that for treating fish, right? Control. After every harvest, dry and lime pond. Very, very important. Because when you harvest fish or shrimp, uh, there could be a possibility that there are some parasites in the water. Adult fish is sort of immune to the parasite. They may not show clinical signs. So it's best to clean the pond properly, sanitize the pond or the tanks, right? Treat the broodstock if you're using broodstock for breeding purposes. Keep young fish separate from the adult fish. Quarantine new introductions because the new introduction could be carrier of the virus, not showing any clinical signs. And you can treat water if you are using water from a stream that has fish with infection, the same infection, then you can filter your water. Use filter bags or gravel filter and things like that. Right? So very, very important, these treatment aspects, these treatments not only used for white spot, but also used for most of the ectoparasitic diseases. I'll explain to you a little later as we go along. So that was Icterus multifilis causing white spot disease in fish. Right? Now the white spot disease in shrimp is different. It's caused by a virus. So white spot in fish is only caused by parasite. All right, let's go on to the next ectoparasitic disease. It's called chylodeniasis. All right, This is the shape of the parasite. It's called Chilodinella. And I will explain to you the different features or characteristics of the parasite. You can see it has a cytosome. That's the feeding apparatus. It has ciliary rows on both sides, on the left side, on the right side, ciliary rows, 4 or 5 ciliary rows on either side. And there's a macronucleus. So it's no longer horseshoe shape, it's round in shape. And it has vacuoles. You can see the vacuoles in the cytoplasm. All right. This is an electron micrograph picture of the parasite. You can see the cilia. All right. Now, it has been described as oval heart shape. But when you see under the microscope, it's usually round. I will show you some pictures. It's granular cytoplasm with vacuoles. A macronucleus is large, 1/3 the body size, and it's about 40 to 70 in width. So it could be smaller than your ectotherius multifilus that causes white spot. And it produces cysts under adverse conditions. As I mentioned earlier, it has left and right ciliary rows on both sides, cytosome, and the marine equivalent. Just like for the fresh water, for Ichthyotherius, we had cryptocrayon. And similarly, for this parasite, there is both fresh water is Chilodinella and marine is Brocanella. Now, this is how it looks like under the microscope. When you make a fresh smear, as I showed you earlier, you just take a scraping of the epithelium or mucus from the fish and you put it on the slide. You view it under the microscope. This is what it will look like. And you can see the ciliary rows on both sides. You can see the cytosome here, and you can see the vacuoles there. Now, the macronucleus is not that clear, but this is, I can imagine this is the macronucleus. It's very large, but 1/3 the body size. And you can see a few hundred sometime in severely infected fish. Now, this is mucus scraping from the gills, right? So you can see all these are the parasites, and you can make out the macronucleus, right? If you go under higher magnification power, you can see the ciliary rows on either side. Okay, you can see the gills. There's the gill filament and all this mucus scraping, and then you can see the parasites. So you can see in fish, clinical signs, in fish infected with this disease or this parasite, you can see ulcerations, this shallow ulcer beginning, right? Now, if it's a severe case, I'll show you. And this is when you do histology, you can see the mucus sloughing and you can see the parasite in the mucus sloughing here, round, right? Over here, right? Right. So that is the Chilodinella causing ulcers. Okay, I'll take you to another one now. It's a long lecture with a lot of parasites, so I'm trying to cover them as quickly as possible so that I can finish the ectoparasites today. So the next one is trichotillomaniasis. That's the name of the disease. The name of the parasite is Trichodyna. It's a Trichodyna complex, a lot of species, a lot of genus within this category. And it's a very beautiful parasite. You can see it's round in shape, and it has spines like that to attach to the body. It looks like, from the side, it looks like an inverted bell, I showed you the picture earlier. That is from the bottom, all right? And scanning micrograph, you can see how the parasite looks like. And you can see the cilia all around the body. Looks like a hat, isn't it? You can see, very beautiful animal. Look like, if you look from the bottom, it looks like a flying saucer. So you can see when you make fresh smear, you will see them flying around under the slide if you have enough water with it to view. So there you are. It also has the macronucleus, horseshoe shape, but it's difficult to see under a normal microscope. You can only see it under the electron micrography or under a research microscope. Here again, so you can see it's bell shape, and you can see the spines here underneath. So it's circular in shape, bell shape from the side, horseshoe shaped macronucleus, move rapidly with the cilia all around the body, and circle of hooklets I showed you there. It can up to go to, it's very small, can go up to 100 micrometer or microns. Very beautiful. As I said, you can see the cilia, you can see the hooklets clearly. Now, this picture is taken under a research microscope. You may not see it as clear as this under your normal student microscope. Here's another picture. A horseshoe-shaped macronucleus. And this is how the histology picture looks like, how they infect on the gill filaments. And you can find a few hundreds of them. All right. And the clinical signs will be the same as I described to you earlier. There'll be excessive mucus on the gills, excessive mucus on the body surface of the fish, except it will not cause the white spot or the white cubicles on the skin and the gills. It will just cause excessive mucus. And in the gills, there'll be hyperplasia. Everything else is similar. Fish will come up to the surface to get oxygen. Fish will die in the night when the oxygen level are low, and they will gather at the inlet where the water is coming in because there's higher oxygen level, right? Everything else is same, and the fish will show clinical signs like swift movements in the erratic swimming in the water, right? Okay, and the treatment same. Just like for white spot disease, you use formalin, you can use methylene blue, and the control measures are the same, right? All belongs to ectoparasitic group, same treatment, almost the same clinical signs. All right, we go to the next one, ichthyobodo. Ichthyobodosis is the disease. The parasite is ichthyobodo. All right, very small. This you only can see under magnification of 400 or the lens 40. Very small. All right, they have a flagella. They are bean shaped in bodies, about bean shaped. And of course, previously, some of the earlier books may call it Costia. You may see the word Costia. This is the earlier name, all right? And then the taxonomy changed, so they gave it a new name, Ichthyobodo or Ichthyobodosis, the disease. So its kidney are bean-shaped or oval. When attached to the skin, appears patch-like form, non-motile at high temperature. They start dying at high temperature. Form cyst in adverse condition. The life cycle is 1 to 2 weeks, depends on the temperatures. And higher temperatures, all protozoans multiply faster. This is how it looks like in histopicture, histology section of the skin. And you can see it's pear-like shape. And that's a flagella. that attaches to the epithelial cells on the skin, or it could be on the gills, right? All right. The next one is epistylis, right? It looks like a cup, inverted cup, and there's a macronucleus. There is cilia on the top, all right? Uh, there's vacuoles, contractile vacuoles. There's a stalk. And on the stalk, you can see in the next picture here, there could be many of these parasites. They use the stalks and they attach to the body. Again, in this case, you can see, all right, this is the top part, right? That's a stalk. And in this case, the clinical signs, you can see cotton-like, cotton wool-like attachments on the fins or the body, right? So this is all epistylis. It can be confused with fungus. So you got to do a smear. If it's fungus, it will not show these sort of structures, right? And it will show, if it's fungus, it will just show you the hyphae, right? So you can see in this case, there are other infections as well, secondary infection with bacteria. We call it fin rot. So there is fin rot. You can see there is congestion here, and the fin is undergoing necrosis, right? And these are the epistylis. Usually, when there is high organic load in the water, you will find epistylis. It's sort of a secondary infection when the water is not clear. All right, we finish the protozoan group, right? I'll quickly take you to the next group.
Prof. Sharif, is it okay if I record? Prof. Sharif, is it okay
Yeah,
if I record?
yeah, go please.
All right.
I thought you were already recording.
Yeah, sorry, I missed.
All right,
Thank you.
okay. So the next group, okay, the first group, remember, there are three groups: the protozoan, the crustacean, and the monogenetic trematode. So I'm on the next group now. So the parasites that I described to you in the protozoan group, there are many, many more. I did not, we cannot cover all. There's so many. So I just gave you highlights of some of the commonly found protozoan parasites. So I'm going to the next group now, the crustacean group, right? All right, before that, my quote for this lecture, right? It's very, very important. The first quote you remember, think carefully what you're going to say, and it should pass three gates. All right, I think you know what I'm going to I said, so I won't repeat that. So my next quote for this particular lecture is, it is not the strongest species that survives, and it's not the most intelligent, but the most responsive to change. What does all this means? What does the quote means? Imagine, we are talking about parasites now. And you saw how the parasites morphology changes. You saw the trichodyna, it has a different shape. You saw the white spot, it has a different shape and different morphological features. All ectoparasites, I will show you, has different morphological features. What they do, why they all are different. They have adopted through evolution, they have adopted to the environment. At the end of this lecture on ectoparasites, you will understand better how parasites adopt to the host or adopt to the different environment. Now, this is a very important code I would like to share with you. Even in our daily life, you could be very intelligent. You could be very strong, big size. But if you don't adapt to the environment or adapt to the changes, you cannot survive. Let me give you an example. You start working in a clinic, let's say. There are 3 or 4 other doctors. And you want to go use your own method. You don't listen to your colleagues, right? You are a little bit arrogant. You feel you know better than them, you know, and you don't share knowledge. You don't work together with them as a team. It will not work. You will have to, they will sack you. All right. Even in the faculty or even in the class, you are a big class. You should work as a team. If you cannot work together with your colleagues, you won't be very successful. So that's why you must adapt to the change wherever you go. You work in a clinic, you work as a salesman, sales agent for a feed company, or whatever it is, drug company. You have to adapt to the system, the practices of that company or of the clinic or whatever it is. Even in your house, if you cannot get along with your siblings, with your parents, that's not very good. So this quote is very, very important. You should have to look at it in many aspects. All right, let me go on. So crustaceans. Crab is crustacean. Shrimp is crustaceans. How come there are parasites, crustacean parasites? Yes, there are crustacean parasites. Okay, this is just an example of what we call it lernia. This is the head region, and that is the body part. It's quite long, 22 mm, not micrometer, so not micron. This is mm. So it's quite big, like a matchstick. You can see it with your naked eye. I will show you. And you can see it's a crustacean. You can see how it has adapted to the host and why it becomes an anchor-like structure. Anchor is a modification of the crustacean so that it can anchor in the body. Right, let me show you. All right, the other parasite in this group crustacean, I'm going to deal with 2 of them, is the Uglus group. And again, you can see here cup, suction cup, we call it. There are 2 suction cups, and it sticks. The suction cups are just like you stick something to the glass wall, you use a suction cup. So it has modified the body to have these suction cups so that it can stick to the hose without dropping off. All right. So crustacean parasites, it has 2 body divisions, the cephalothorax, head, thorax, and the abdomen. Cephalic region, the mouth part is specialized. I showed you how the anchor part of the worm. So it's specialized for piercing or sucking. And the thoracic appendages are often reduced, 4 pairs of swimming appendages. I'll show you again here. So you can see the swimming legs, very rudimentary, reduced in size. All right. So we go on. So with the anchor, let's start with the anchor worm. We call it anchor worm because it's anchor shape. It's called lernia. Common in cyprinids, especially in cyprinids, the carp group, right? Koi fish, goldfish, these are the cyprinid group. Female, this is the female parasite. it exhibits modification of cephalothorax. So this has changed in shape and embedded in the body. Only the female penetrate the host. This is the female. The male drops off. You can see how the anchor goes underneath the epithelial cells, in between the scales. All right. So you can see the body part is hanging outside, and it has a Y shape at the end. These are the egg sacs at the end here, egg sacs. So they will hatch. That's how they reproduce. So again, clinical signs almost the same as the Ichthyophilus multifilus infection or the white spot. They are all the ectoparasitic group. In this case, you can find localized hemorrhagic lesions because they penetrate into the underneath the scales, anorexic, acute mortalities in fingerlings, loss of weight in older fish, if there are many. Secondary infection, you can get secondary infection by bacteria, by virus, by fungus. And that's it, the cephalic region. Swimming lake I showed you earlier. That's the Y shape at the back, and these are egg sacs. And these are the stages of life cycle. Nucleus, remember crustacean, the crab, all the other crustaceans, the shrimp, they're all crustacean group. And they go through this sort of life cycle. The nucleus, of course, the shrimp goes through many, many stages. And this is only 3 stage, nucleus, and then 5 stage, copypodid stages. So it is at this stage that the parasite penetrate the host, not this stage. This stage develops when it is in the host. When this has penetrated the between the epithelial cells, between the scales, then the anchor will develop so that it doesn't drop off. You see, it's big size. If it doesn't have the anchor, it will drop off with the swift currents. So you can see how the adaptation through evolution, each parasite has adapted differently to the host. So this is 5 stage, this is 3 stage. The total takes about 14 days, 2 weeks. And that's why the treatment I will explain to you later takes very long. And that is the stage it penetrates underneath the epithelial cells, underneath the scales. So you can see its sharp claws. And of course, it also produces an enzyme to digest its way underneath the tissue. You can see here fish heavily infected. This is bighead carp, one of the carps, heavily infected with this parasite. There were more than 400 parasites on this one single host. And they feed on the host fluid, body fluids. Heavy infection. Why green in color? Because of the fungus from the tank. So fungus also gets infected, infect the fish. And you can see all this like matchstick. So those are the parasites. And in some cases, you can see the egg sac at the back here. Yeah. Here, Y shape at the back here, the egg sac. So they are on the body for about 3 weeks. And they keep laying eggs, hatching. And eggs hatch into nucleus, copypod stage, and then they infect the host. Only the female infect the host. The male will die off after copulation with the female. Here, goldfish is also the carps. You can see many of them. And if you do histology, you can see this is the scale with the sharp ridges. And then you can see this cross section of the anchor. See another cross section of the anchor. You can see here, because the anchor has a lot of branches, remember? So this is another branch cross section. And you can see a lot of cellular response from the body. All right, lot of inflammatory cells. Here again in the eye, so they can even infect the eye, and then they damage. This is the lens. They damage the lens. All right, and the fish will become cataract. We have a cataract. So, how to treat crustacean ectoparasites? Uh, they use pesticides in agriculture. And one of them is diptrax. We call them organophosphate. So you can use the same thing, organophosphate, at 0.25 ppm. Depends, you must watch carefully the, the active ingredient. The active ingredient, work that out. It could be only 40%. So you work that out and to have 2055 ppm. Weekly application for 4 weeks. Remember, I told you they lay eggs for 3 weeks. So that's why the treatment is long. If you start on Monday, apply for the next 3 weeks at 0.255. Only then you will see the reduction of the disease. All right. It's nice to show you, just to share with you. If you go to Taj Mahal, you find a lot of people selling these marble plates, all right, very decorative pieces, very nice. All right, the next one is agallosis, right? I will quickly try to finish this. So you have the suction cups there, the eye, proboscis. This proboscis is used to prick or to prick the skin to absorb the body fluids, right? And you can see the thoracic. So you can see the adaptation of the parasite. Linear, different structure. It modifies the cephalic region. In this case, it becomes like a plate. All these are crustacean group. It becomes like a plate, right? And it adapts with the suction cups. So this is all adaptation, and that's how the parasites survive. All right. So this is called also fish lice or kutu ikan in Malay, Agalus species. Dorsoventrally flattened, covered by carapace, like the tortoise shell, all right, carapace, forming a shell over the thoracic and abdomen. You can see the shell here. Retractile stylet, proboscis like mouth here. 4 pairs of swimming appendages, you can see there. 1 pair of maxillae modified into large sucking discs for attachment to the hose. This one here. Remember I told you how you stick something to the glass wall? So they function exactly like that. They stick to the hose. Actually, it's difficult to remove them from the hose. You must use a forceps, otherwise it's difficult. There you are, the parasite I showed you earlier as well, the appendages, the suction cups, and it has eyes, and the carapace. All right, so you have seen that picture. So it's quite large, you can see with your naked eye. There you are on the fish hose, quite large. Of course, there are different species, some are smaller. You can see how the camouflage as well. Sometimes they almost resemble or camouflage with the skin color. You can see the eye spots. And these are smaller species, and there are many here. This fish actually was from Country Height nearby UPM, and they sent to our lab, and it was infected with hundreds of agalus. So it goes through different, it's not similar to the linear. It goes through different larval stages and then it becomes a mature adult. And its life cycle is a little bit longer, right? And this is how the adult parasite will lay eggs on anything, any substrate. It could be a pipe, it could be a stone, at the bottom of the tank or at the bottom of the pond, right? The pathogenicity, because of the stylet, it pierces the whole skin, so it's harmful for the small fish. And as I said earlier, you can get more than 400 parasites per fish. Fish becomes restless, reduce food intake, anorexic. So all clinical signs for ectoparasitic infection are very similar. Increase of mucus cells, inflammation, aggregation of lymphocytes, if you look at the section, necrosis, of course, ulcer, hemorrhages. You can see here, quite badly infected, hemorrhages, right? This may later become ulcers. All right, it's 9:55. Let me go on to one more. I'll take only 3 minutes for that. Now, this place is in Morocco. I used to travel quite a bit to give lectures. So this is Morocco, one of the palace walls there. Quite interesting. Morocco is next to Europe, and majority of the people are Muslims there. All right, isopod. You may see in your practical class another crustacean parasite known as isopods. You can see how large it is, and it is usually found in the mouth part, oral cavity. You can see one sitting there. So it's almost blocking the passage for the fish to feed. So the fish becomes emaciated. And it's also found on the gills. All right, very nice covering. The operculum covers and they lodge at the gills, the mouth or the gills. All right. So when you're doing practical class, look out for them. You can see here, quite big, isn't it? A big size. You won't miss them. So that covers the crustacean group. And I have one more to talk to you, the monogenetic group. So it's been almost 1 hour. Let's take a break. Maybe I can give you maybe a 10 minutes break. All right. We'll come back at 10:05. Okay. 8 minutes break. All right. See you.
Hey, Prof.
Hi, Diana. You there? Yeah. Are we on the full screen?
Yes, Prof.
Okay. Thank you. Maybe I'll come back around 10:05.
Okay. So students, I've already told the class rep that the test one will be on the 22nd of November. It's in on Monday. So we will start at 5 with I'll give a bit of briefing, and then you guys can log in Putra Blast at this specified time. So usually for tests, you guys have to open WebEx as well, right? Is it true? Is it right?
Yes,
So while you
doctor.
guys, all right, so while you guys answer the test, will be like this in the WebEx. Okay. Um, MCQ, MCQ, one answer only, right? I mean, like each question, so just choose one right answer. And then I think two, two short answer, question. 40 questions in total. Very, very easy. Please read up. I told Fahad that so it will cover from the first week until the presentation on Monday. Um, I do not know. Should I include this lecture as well? I will let you know. If so, lecture from Sharif will be only two questions if I decide to include. Um, the questions are ready. Um, ready. Just waiting for Monday. Okay. All right. That's all I want to say.
Doctor, I have question, doctor.
Yes. Is it for me or
Uh,
is it for Pro
yeah,
Sharif?
untuk Doktor Diana.
Oh, okay.
Doctor, like during test 1 nanti, can we go back to previous question? Like which macam nak
Yes,
skip-skip
it's a free
soalan?
navigation. I already
Uh,
studied free navigation, so you
alright.
can and change your answer.
Okay, doctor.
45 minutes only, yeah. Um, 40 question, 45 minutes. I think you will have enough time, no problem. Because I've run through it a couple of times. Yeah, you have enough time. Okay? Do you guys want more time? 45 minutes not enough. Should be enough. I do not want to hold you guys for too long. Unless you guys want to do it next Monday, then we have more time because I'll just give very short briefing, like a bit of briefing. I mean, Amalila practical online and then briefing. And then if you guys want to have the test earlier, so you have more time, next Monday can. Or the other, maybe.
No,
Okay, no, okay. All right. You guys want to study. Okay, no problem. Okay. Laju je cakap no. Anyway, that's why senang je lah. MCQ. Tiga mata je jawab. Ha. Soalan Pro Syari dulu lagi susah tau. Ha. I dah baca dah soalan ni. Syari tak dengar. Okay.
back. It's 10:04. Maybe one more minute. I hope I'm not too fast. I got to finish quite a lot. We'll give them session for question and answer at the end of this.
Yeah. Prof.
You have the numbers there, or they are still online, or
Yeah, they are still online.
okay. Okay.
We can start.
All right. Welcome back. It's 5 minutes past 10. So I hope you had a short break, bathroom break, or whatever it is. I went to get my drink. Right. So we have covered the protozoans, the crustaceans. Again, as for the protozoans, there are many other species. Similarly, as for the crustacean parasites, there are many other species. I just covered two common ones. Next, we'll continue. We'll talk about the monogenetic trematodes, or class monogenian, they call it. All right, monogenia. Okay. Now you have studied the liver fluke, and you can see how when the parasite is in the body, how it looks like. Looks like a leaf, isn't it? Flat leaf. And how it has adapted when it becomes an external parasite. You have not studied in your DVM. This is the first time for parasitology. You have not studied monogenetic trematodes. So you can see how they have adopted to the external, as an external parasite. All right. Now, as I said, I always give quotes when I start a lecture. Successful people don't reach their goals alone. All right. Very, very important. As I mentioned earlier, you are a class. You are like a family. You should work as a team, not think always of yourself. You are not successful until you help others succeed. All right. So I was a lecturer in UPM. Of course, a lot of students graduated, did their masters, did their PhD. All right. Well, Dr. Hassan was my student. Diana was my student. Okay, she just finished her PhD. Right. So you must help each other. You must work as a team. Right. That's why even if you're working in a clinic or anywhere for that matter, at home, you should work as a team. All right. Help your younger siblings. Okay. All right. I'll go on. Now, this is the comparison between digenean and monogenean. Here onto the left-hand side, you have the digenean parasite. Example, the liver fluke. And here you have the monogenia, or the monogenetic trematode. And you can see here, the oral sucker is very clear. And the ventral sucker, oral and ventral sucker, very clear. Here it's been modified. Why this oral sucker and ventral sucker is well developed? Because they have to stick somewhere in the body, But in this one, it doesn't need that, because it has a better adaptation mechanism. They call it the opihapta. down here. So instead of the oral and ventral sucker, it has this mechanism of anchoring on the host. Remember, it's an external parasite, right? So it has two large median hooks, and it also has hooklets, marginal hooklets, right? It could go up to 6 pairs or 7 pairs, depending on the species. And I'll show you a few other mechanisms, how they adapt to this anchoring themselves to the host. So you can see it's almost similar. The body shape is trematode also, all right? And this is the major difference, okay? So class Monogynia, most characteristic structure, opisthepta, as I showed you earlier. Anterior small, less developed suckers. Suckers, you can hardly see them. It's hermaphrodites, most oviparous. That means they lay eggs. In some, they have uterus as well. They develop fetus in the uterus. Okay, they develop the larvae in the uterus. All right. The monogenetic and digenetic, the difference. Just now you saw the morphological difference. Here, the monogenetic trematode, direct life cycle, no intermediate host. The digenetic, remember, the liver fluke has to go through the snail, the scarae, and maybe the birds, and so on. So complex life cycle involving one or two intermediate hosts. And this one has the presence of the opisthepta, I mentioned to you. This one has a better mechanism for the distinct internal, because they are inside the body. They need oral and ventral suckers. And these are endoparasites, and these are ectoparasites. So that's the difference between monogenian or monogenetic. The class is monogenia, right? Otherwise, we call them monogenetic trematodes. All right, here is the parasite. And you can see the median hooks. I'll show you the median hooks and the marginal hooklets. It has eye spot, all right? It just looks like a fluke. Better picture of the median hooks, 2 piece, 1 pair, and the marginal hooklets. And here to show you the different, I told you they are oviparous. Some, they have the embryo within the body. So this is one very special parasite. It is 3 generation, the adult and the younger one here. In the younger one here that's developing, there's another younger one in there. So they call it 3 generation parasite. And these are quite rare, I think, among animal kingdom. All right, they are, or among the invertebrates is rare. Okay, again, you can see the marginal hooklets here, and the main hooklets, or they call it the median hooklets. Let me, my battery is going out. Okay. I hope I didn't lose the link. All right, my computer was adapting to the change. All right. All right. I hope I can go on. I think due to the... Okay. So you can see how, so this is what I showed you earlier. And you can see the other parasites among this monogenian class, they have different structures. These are suckers. See, instead of marginal hooklets, they have adopted different mechanism. And here, different mechanism, right? There's so many. Again, the marginal hooklets are very small in this case. Here too, very tiny, you know. And you can see here bigger sucker. So there are many, many variations of the monogenea class. All right, here is what you will see if you do a wet smear. All right, just do a scraping of the mucus or the gill surface. You will see these sort of parasites. This is sort of a bent, all right, bent. It should be straight, but just to show you, you can get in different forms when you do a smear. And you can see the median hooks very clear in this case. You can see the eye spot here, right? This is the anterior part, this is the posterior part. Electron micrograph, you can see clearly how the marginal hooklets anchor onto the body. These parasites are found on the gills, on the skin. Some are specialized more on the skin. Some are found more commonly on the gills. You can see how they anchor. See the anchor mechanism, the marginal hooklets, right? And these are the median hooks, very large. They have a link, ventral bar link there. And this from the bottom, a picture taken from the bottom, you can see the marginal hooklets and you can see the median bar. All right, the life cycle. No intermediate host, remember? So the adult on the gills here in this case, and they come out as they hatch, the ovi, remember oviparous? So they lay eggs, the eggs hatch, and then they find the host. Very simple, no intermediate host. So the life cycle, free swimming larvae emerge from the eggs covered with ciliated epithelium. You can see the epithelium, all right? So that's how they find the host, so that they can swim. Attached to suitable host, eggs develop within 1 to 4 days, depending on temperatures, as I say. All right, so the egg hatch, what day, how many days. Larvae live only 6 to 24 hours. So you can imagine if you have an infection in the aquarium tank, you don't have to use chemicals. Just remove the fish. Of course, you got to treat the fish. and if you want to make sure there's nothing left in the water, just leave it empty for 2-3 days. They will disappear. All right. Now, there was an external examiner for the final year from, I think he was from New Zealand. He was a little bit crazy about fish parasites. Maybe he has done a little bit of work on fish parasites for his coursework. And he asked one of the students, what's the difference between Gyrodactylus and Dactylgyrus? So here you can go back and look at it. Okay, I won't go through with you. So remember I showed you the Viviparous, the 3-generation parasite. There is Gyrodactylus, right? Second embryo appear in the first embryo, or perhaps the 16, here 14, you know, just in case, all right? if the examiner asks you some difficult questions like that. All right, the clinical signs. Remember I told you, ectoparasites mainly similar. Blue grayish mucus layer, gill spotted. In this case, because of the anchor, marginal hooklets, there could be some petechiae hemorrhage. Hyperplasia, I showed you. Excessive mucus. Of course, there could be necrosis taking place. All other signs as per ectoparasitic infection. The fish will show erratic movement, ulceration, shallow ulceration, depending on the severity of the disease. They'll come to the inlet, to get more oxygen. If the gills are really badly affected, same. This is a bad case. Ulceration, very severe infection. And you can see the shallow ulcers, right? This is how they attach to the gills. See, this is the gill filament. This is primary lamellae, this secondary lamellae. So you can see, you can imagine there are the marginal hooklets, they attach. That's how they attach. Okay. So all this is part of adaptation to the host for them to survive. Right? So as my quote was, you should also be able to adapt to your new environment wherever you go, wherever you are. All right, so you can see them. This is not small, but when you cut sometimes, when you do sectioning, you get smaller piece, although they could be just as long as this. There are many here, you can see down here, all right? Here, you can see how severe the hyperplasia is. And that's the parasite there, the monogenetic. And that's, imagine that there are marginal hooklets anchoring the parasite. So the treatment, what's the treatment? Same. You can use formalin, you can use Diptrax, you can use malachite green, and you can use, this is for, because they are flukes, you can use the same thing you use for liver fluke. All right. Any questions? Now, I kept a lot of time for answering questions. I went through quite fast, and I realized that I could have slowed down a little bit. Never mind. We have a session because this is not face to face. This is online, and I have all the slides. If you were not clear on certain point, I can show you the slides again. Right? So you can see from my slide presentation here, I have actually removed quite a few slides to make sure that I can cover all the topics, the 3 topics of ectoparasites. So in some cases, I've removed some of the slides so that I don't repeat, right? But rather than me talking, let's listen from the students if they have any questions on ectoparasitic diseases. I'm waiting for
Students,
you.
we ask
Yeah.
questions. Prof. Sharif will only be with us for 2 classes. Please ask as much questions as possible. Ada soalan tak? Tolong tanya soalan.
Ada. Ya,
Okey, tanya.
Go ahead.
hello, doctor. So, today we covered like all 3 of the diseases, right? And a lot of the treatments were similar. Earlier
Yes.
you mentioned during the one for parasites, where you couldn't lower the temperature, you couldn't increase the temperature of a large tank. To control the outbreak. But what if it's a small, like a smaller tank
Yeah.
for
Thank you for the question. Now,
white
your
spots
question.
specifically?
Okay. Keep your second question. Let me finish your first question. All right. Your question is, if I got it right. Now, the especially the white spot, or for that matter, any stress factor, overcrowding, water quality is no good, dissolved oxygen is low, all these can cause stress to the fish. But for the white spot, it was very clear, and most of the diseases, right, when what we call it, when the fish is immunosuppressed, That means, imagine you going and sitting in the fridge or a cold room. You are locked inside the cold room accidentally. How will you feel? You feel very cold. All right. Now, some of you have been to cold countries or temperate countries during winter. Wow. Yeah, you feel you shouldn't be outdoor. Immediately you want to rush back to your room where there is heating system and so on. Similarly, the fish, even by 1 or 2 degrees Celsius, lowering of temperature can cause a lot of stress to the fish. And if you measure the cortisol levels, they will increase, right? Now, I have a presentation somewhere to show you what causes stress in fish, what triggers fish infection. Actually, in most cases, is the stress. So lowering of temperature during cooler weathers when it's rainy season, you will find more diseases. Or during drought season, you will find more diseases. When there's a change of climate in Malaysia, rainy season, we don't have winters. So we have rainy season, we have dry seasons. So during this change of seasons, you will find more diseases in aquaculture practices, right? So you can increase the temperature in tanks, in aquarium tank, by using a thermostat, right? In ponds tak boleh. Don't talk about ponds. You cannot do much because they're so large, 1 hectare, 2 hectare. Only in aquarium tanks or in a hatchery tank. Yes, you can do that, right? I will try to pick up the presentation if possible. I will show you how people use overcome low temperatures, right? Okay. What was your next question? I hope I answered your question.
Yes, you did. My next question was after like reading a bit about white spots, I came across something called salt baths for the freshwater species. So I wanted to ask, since you mentioned that excessive stress on these fish species like makes the incubation of the white spots even worse. How exactly does putting the fish in a salt bath make it better?
I really didn't get your audio is Diana, did you get the question?
Prof. Sharif, her question was, for treatment of ectoparasites,
Yeah.
how does salt bath help treatment? How does that work?
Oh,
Salt,
okay.
yeah.
All right. I think it will be covered under treatment, chemotherapy. Uh, Imagine you going to treat fish, short bath, we call it dip, DIP, dip. And the other treatment is bath treatment. Bath treatment, you put a lower concentration. Let's say malachite green. Give you an example of malachite green. Let's say malachite green, you use 2 ppm for a bath. That means you put it there and leave the fish there. Of course, for malachite green, no, sorry, for methylene blue. Don't use malachite green as an example because malachite green is very toxic. Methylene blue, I'm talking about methylene blue. If you use methylene blue, let's say you use 2 ppm, right? And you leave it there, and every alternate day you add another 1.1 ppm. Okay, so it's a long-term bath. That's what we call it bath. For dip, you use higher concentration. You may use 4 or 5 ppm, but the duration is short. You only treat for 10 minutes, depending on the case, depending on how severe the fish are infected, and whether they can tolerate that high dose. For high dose or for dip, we call it DIP dip. For dip treatment, you use higher concentration of drug, but you are also going to stress the fish further. The fish are already sick. They are already under stress. So I would not recommend a short-term treatment. I would recommend a long-term treatment. Right? I hope I answered your question. It's just that the difference is you use a higher concentration, but shorter duration. For bath, you use a longer duration, lower concentration. So the fish will tolerate and survive. Okay? That will be covered. I think Diana or someone
Yeah,
will cover.
in
Yes.
chemotherapy.
Yeah.
Afika, did Prof. Sharif answer your question? Do you need
Yeah,
more
it did.
clarification? Okay. So Prof. Sharif, I will read two questions on the chat box.
Yeah.
First question, ectoparasite freshwater, can they survive in marine and brackish water?
Very interesting question. Thank you for asking that question. Remember, I said in one of my slides, you can use sodium chloride, your table salt, to treat for ectoparasites. So, same thing. That is for freshwater fish. So, what you do for marine fish, you reverse the salinity. That means you put them in fresh water. Of course, you cannot do that for a long duration. That is called dip treatment, DIP, drip short-term treatment. You put them in fresh water, the marine fish, you take them and put them in fresh water. Of course, sometimes it's not practical. Let's say if you, I think most of you must have seen cages, how fish is cultured in cages. And to get fresh water is sometimes difficult. Of course, for juvenile fish, for small fish, you can bring a few buckets of fresh water and put them for 3 minutes or 5 minutes in fresh water, and the parasite will die or the parasite will leave the body because of the change in the osmoregulation. Remember? Marine to freshwater. It changes. The whole thing changes. Same thing. Freshwater, you use salt. For marine fish, you reverse the salinity. You bring them into freshwater for a short duration. Don't keep them for 1 hour, they'll be all dead. Just 3 to 5 minutes, you observe, you watch, you do a trial treatment first to see whether they can tolerate or not. 3 minutes, you use 5 fish first. They can tolerate 3 minutes, then you can treat 2,000 fish for 3 minutes and then put them back. All right. So it's labor intensive treatment of dip because you quickly got to reverse whatever you're doing. Let's say you're treating for formalin or methylene blue, or you are doing a dip treatment. After the 5 minutes, you got to remove the fish back into their tank. So it's labor intensive. You got to move fish around. It's very stressful as well. So dip treatment is only used for healthy fish that are new consignment. You quickly dip them. They look okay. Okay, quickly dip them and put them in the tank. But for sick fish, try to avoid dip. Use bath treatment. I hope I answered your question.
Muhammad Rizwan, you, are you okay with the answer? Never mind, I move on to the next one. Yeah. Next one, I think question by Yuki was answered by Prof. Sharif in the previous question. Third question, Amar asked if there is any common fish ectoparasite that can transmit zoonotic, any zoonotic ectoparasite in Malaysia fish.
All
Yeah.
right, good question. Thank you. No, none of them, as I know. All right, they are very fragile parasite, especially the protozoan. Now, when you are having the practical class to examine fish for protozoan, Diana, have they done that or they're going to do that?
They are going to do that.
Okay, so you will see one of the instructions will be keep your fish wet all the time while you're examining for ectoparasites. if the fish becomes dry, all the protozoan parasite will be dead. So the fish that you buy in the market is all dead fish. All the protozoan parasites are dead, gone. And Linear, of course, the crustacean parasites cannot infect you. It's not zoonotic. Monogenetic is not zoonotic. No. And the other thing is that even if you get fresh fish, you cook it, our cooking, the way of our cooking, you won't get infected. There's nothing zoonotic among the ectoparasites. Endo, yes. I will talk to you about endoparasites. The helminths, yes. Okay. Other
Amar, I hope.
questions?
Yeah, there's another one question. Good morning, Prof. Sharif. Earlier June this year, there were sightings of white spot lesions on the white tip reef sharks in Sipadan sea waters. May
Okay,
I know
okay.
what is the best solution way of treating shark population
Yeah.
since they are not
Okay.
in an enclosed setting?
Yeah. There was a news item. I responded to that. People asked me what was the story behind that. I don't know whether I can gather that I have saved it or not. But it was, we suspect, because of the changes in the weather, climate change, it's a little bit warmer. Our seas maybe half degree or 1 degree. I'm not sure how much, but that particular region, they said the water was temperature was a little higher, and with that sort of record or report of higher temperatures, there has been reports of fungal infection. So I have that. I hope I can find that and send it to Diana. That was the story. So it was not really white spot. It was fungal infection that perhaps looks like shallow ulcers could have made it look like white spot. Right? Hope I answered your question.
Dwayne, you okay? All right. So his next question, how do we treat the fungal infection since it's in the sea?
Yeah. Actually, I think either Diana or Dr. Hassan will be covering with you fungal infections. They are also ectoparasites. They are also similarly treated with formalin, methylene blue. You can also use salt, common salt. All right. Similar treatment like for ectoparasites because they are external, right? Hope that answers your question. Okay. Even on eggs, you can get fungal infection on eggs. Of course, normally if the water quality is bad. No, the thing is that first question to ask for any treatment, very, very important. I'm sorry I didn't highlight this. First question to ask, what triggered the infection? Under normal circumstances, the fish has a good mucus layer. Even take our body for that instant. Our mouth, it has saliva. Our eyes, it has tears. Now, the tears have fighting mechanism. If something goes into your eye, automatically there'll be excessive flow of tears. Why? To dilute whatever is causing the irritation. And the tears has chemicals in it, right? To get rid of bacteria or whatever it is. Even the saliva, same thing, all right? It has a mechanism, chemicals in it to protect. Similarly, in fish, the body coating, the mucus layer has chemicals in it, antibodies and so on. Normal circumstances, nothing will happen. So, what triggers the disease? Let's say your fish is dying of white spot, for example, or trichodyna. For example, first find out what is the stress factor. Lowering temperature? Okay. Make sure you raise your temperature if possible. Overcrowding can cause stress. Maybe the farmer becomes too greedy. He, instead of putting 2,000 fish, he puts 5,000 fish. He wants to make more money. But you remember that any organism has its tolerance level. Beyond a certain tolerance level or adaptation level, he will not survive. He'll become under stress, and that's the time the organisms, now white spot, Lernia, monogenetic trematode, if you go to the wild population in the rivers, you can find them, but in very low numbers. Catch a fish from the wild, do a scraping, you will find monogenetic trematode. But they don't cause disease. Why? Because it's a balance, natural balance. Because the fish can protect itself, right? The fish doesn't allow the parasite to multiply to that level to cause it to die. So we must look at the triggering factor. What is the stress factor that triggered the disease, right? That caused the disease, that caused the host or the fish to become immunosuppressed. When you are under stress, now I normally give a lecture on stress. When you are under stress, your cortisol increases. And when your cortisol increases, your antibody production goes down. And that's when you get sick. Your throat has bacteria that can cause throat infection, that can cause sore throat. It's there. So if you don't sleep well for a few days, if you don't eat well, now in teenagers like you all, your boyfriend doesn't talk to you for a few days, or a girlfriend doesn't talk to you for a few days, you get sick, you get flu, you get cough, because you are under stress. So look for stress factors when making diagnosis for fish. It's not just the parasites. You also have to remove the stress factor. If there is overcrowding, reduce the stocking density. If the water quality is no good, there's not enough dissolved oxygen level, increase, do something to overcome the problem. So it's not just looking at pathogens, it's also looking at the environmental factor. Remember during haze, when there is a lot of haze, a lot of smoke in the air, the asthmatic patients get sick, they have to be rushed to the hospital to give an oxygen and so on. So that is stress. So look for stress factors and try to remove stress factors before even you treat the fish. Okay? So that is part and parcel of diagnosis and treatment. Look for stress factors, overcome the stress factors, or avoid stress factors, reduce stress factors, minimize stress factors. Okay, I hope I've answered the question. Other questions?
Other question is, are there any other ectoparasites that have fluctuated due to climate change?
I wouldn't be able to answer that because climate change is something new and there are very few publications on climate change related to parasitic adaptation or fish becoming more. Of course, in Malaysia, it's very clear during the climate change, in the sense that during drought, you can get more severe infections, and during rainy season, more severe infections. There's not much yet, besides a few studies. Yeah. Thank you for the question. It's interesting how climate change is going to affect aquaculture. People have started looking into it, right? Interestingly, you asked that question. Tomorrow, I'm going to Gold Coast. Gold Coast, not in Australia. Gold Coast in Sepang area. Sungai Pilik. I don't know how many of you know Sungai Pilik. To look at farms where we can put probes, dissolved oxygen probe, pH probe, ammonia probe, so that we can get monitoring of the water quality. Sitting in our UPM lab, all data will be transmitted through internet, and we can see the changes in water quality. And then we can advise the farmer, okay, you have a problem now, your pH has changed or your salinity has changed. Do something about it, or else the fish will be under stress. So we are doing that project now. We are going to start that project. We have got funding for that. All right. I'm working with the engineering faculty colleagues. Yeah. Thank you for the question.
On the first lecture, I actually introduced them to IoT, Internet of Things, so they would
Okay,
have
good.
some idea on what.
Yeah. So we are doing exactly, we are doing IoT, and we will be monitoring the water quality parameters. We have got about 100,000, I think, for that project. Yeah.
There's another question, Prof. So Mira said she read an article that says fish with ectoparasites rub themselves on shark skin because it acts as an exfoliant and help to remove the parasites from the fish. Does it affect the sharks in any way? Can they get the parasites from the fish?
Sorry, I
Oh,
missed.
okay. I repeat the question. Can you hear me, Prof?
I can hear you. Yeah, yeah.
Okay, okay. So she asked, fishes with ectoparasites rub themselves on shark skin
Yes.
because the skin acts as exfoliants. So
Yes.
can the shark get the parasites from the fish?
Okay. The question is now there is a symbiosis. All right. Some fish, they always go along with the sharks, and they feed on the shark parasites. Okay. And even, of course, whatever they find around the shark body, right? Uh, this is a natural phenomena. There are many other fish, like the sea anemone. They are hidden under the anemone, sea anemone, this thing. The clownfish, yeah. So the clownfish hides in the sea anemone tentacles, all right? That prevents them from being eaten by the bigger predators. So back to your question. So if the shark eats other fish, you mean, I suppose, uh, the carnivora fish eats smaller fish? Yes. That's how the endoparasites are transmitted. Not the ectoparasites, the endoparasites. Ectoparasites, once you consume them, as I said earlier, they are very fragile, especially the protozoan parasites. They will just perish. They will die, right? I hope I answered your question. Yeah. Helminths, yeah. You can transmit helminths if the carnivora fish eats smaller fish. I will show you in the next lecture how helminths are transmitted from one group to another. Yeah. That's how your cat gets infected when it eats fish. So similarly, bigger fish, carnivora fish, when they eat smaller fish, they get infected. More questions? We have about 11 minutes. Or
Hi,
we can
Prof.
break. Yes, go ahead, please.
Prof, I want to ask, what is the difference between the white spot and also velvet? Because sometimes I can't differentiate between of them. Because when I see my fish, it looks like same, which is spot that white in color. I can't differentiate between white spot and also velvet. How
Okay,
do we can differentiate between
thank
it?
you. Thank you for your question. All right. Of course, the best is to make a smear to see whether you can find the parasite, or if it's velvet, all right, it will show a different morphology. If it's a white spot, it will show a different morphology. But keep in mind, you can get both infections simultaneously. You can get velvet, you can get white spot, you can get monogenetic all together. There's no such thing as just white spot disease. In a severe situation where the water quality is bad, you can get all sort of infections together. Okay, there's no such thing as just one infection. You go to, all right, it could be multiple infection. Okay, I hope I answered your question. Well, of course, to confirm whatever it is, as I said, make fresh smear, look under the microscope. That is to confirm what you have on the fish body. It could be both, but of course you can differentiate from the morphology of the organism. Right?
All right, thank you, Prof.
More
Prof, I
questions?
have
Yeah.
one question.
Go ahead,
Uh,
please.
is that true? Is that true? Usage of seaweed can help to control the water temperature, because some of beta farm they use seaweed to control the water temperature. I want to confirm it with you.
Okay.
Usage of seaweed.
Okay, thank you. You know, TC cake is being used by shrimp farmers and also fish farmers, okay, in some, depending on the condition, depending on how much the experience is and so on. Now, to some extent, the TC cake, it has tannin, I think it also has saponin and things like that. So those act in some way. In some cases, the shrimp cannot molt. So they put that to trigger the molting. And if they cannot molt, they may succumb because they are under stress then. They cannot molt. So they are under stress, depending what environmental temperature is it, or is it the pH is not suitable, or the salinity is not suitable. It could be any of those stress factors. But this, when they add this TC cake, it stimulates, it triggers, right? It helps. The chemicals do help to do what is necessary, what is used to do. But the animal will still be under stress. So it does not increase the temperature. It helps in other ways because of the chemicals in it. All right. I mean, you cannot put something chemical in the water, expect temperature to go up. I think there are very few such chemicals. All right. I don't think in this case, I have not done scientific experiment to show that TC cake will not increase temperature, but I don't think it is the case. All right. It's the chemicals that triggers whatever problem they're trying to overcome. Okay, maybe the tannin, saponin, or whatever it is in there will stimulate the immune or enhance the immune response in the animal, and that helps in that way, not the temperature. Okay, I hope you understand what I said. Thank you for that question.
All right, thank you, Prof.
Thank you. Do they have another class? Do they have another
Um,
class?
Guys, do you have another class later?
At 11, Doctor.
11 o'clock. Yeah, they have another class at 11.
Okay, okay. Maybe one last question. I mean, we have
Um,
6 minutes.
Right, there is another one. Are the ectoparasite species specific? Like fish, can they go to shrimp or other species?
You have specialized parasites for different hosts. Macam the Aglus, the linear group, they are more towards the cyprinates, right? The carp group. And of course, you don't find parasites like Brucunilla and Ichthyotherius multifiliis in shrimp, but you do find the ciliate group in shrimp, like the Epistylis and others, right? Of course, there are some specific, what do you call it, specific parasites for specific host, right? But some of them are very general, like you don't find white spot in shrimp. The Ichthyotherius multifiliis, you don't find that in shrimp. But you find Ichthyotherius multifiliis in all other fish. You can find them in any fish, right? Of course, as I said, the disease is triggered by lowering of the immunosuppression due to the environmental changes. Okay, keep that in mind, right? Okay, I hope I answered your question. There are some parasites that are specialized or specific for specific hosts. Some are very general, are common in any host. I hope that answers your question. All right, I think 56, we say goodbye. I like to thank you all. Thank you, Diana, for inviting me.
Thank you,
And
Prof.
it was great to talk to students. I hope they learned something, not just the technical part, but also the daily life. Remember my first quote and my second quote and third quote, okay? Okay, have a lovely day. Thank you very much.
Thank you, Prof. Thank
Thank you.
you for joining us.
Bye. Bye.
We'll meet you again next week.
Okay.
Thank you, Prof.
Students, if you have any questions, just email me and then maybe I can ask for Sharif later. All right. Thank you. Bye-bye. Assalamualaikum.
Waalaikumsalam.
Thank you, Prof. Thank you, Doctor.
Thank you so much, Prof.
Thank you, Prof.